what is this a picture of? Now, is there anybody here who thinks that this is the Golden Gate Bridge over San Francisco Bay? What is the picture of? Do you know? Huh? What? Well, I can't hear you, but if you said the Brooklyn Bridge, you're right. So this is the most famous bridge in bridge history. And it's a logo for this course because, in effect, we're all bridge builders. At least that's the intent of it. So make believe that on the Brooklyn side are all the advances in contemporary biological and engineering sciences. And on the New York side are all the needs and interests in medicine and in identifying and treating disease. So the problem is there's a huge gap between these two disciplines, which to some extent have almost been moving in opposite directions over the past 30 or 40 years. So this course is a modest effort to try and bridge that gap, and you can visualize yourself as the two men on the catwalk. And so what we have done over the past 17 years is to bring together people who know about a given disease process from a somewhat clinical perspective, and others from a more reductionist, sort of call it basic science perspective. And they present briefly, and we encourage questions and don't hesitate to ask. Now, today's subject is, like almost all the ones we have, ideally suited to be considered as an elegant bridge. I think this is perhaps, this is somewhat subjective, but I think today's subject is the most elegant bridge that's evolved in the past 10 years. And the story really is best illustrated by the two scientists who won the Nobel Prize for this. And so I've divided this into two parts because each one has a brief story which is particularly relevant to young scientists here and elsewhere. Part one is Sir John Gurdon, who, when he was a student at Eton, he scored in the absolute bottom of his classes. And when he eventually went to Cambridge, his professors wrote, "There's no future here in biology. Go be a lawyer or something, I don't know what." At any rate, 64 years later, he was widely recognized as one of the finest minds of his generation. How did that happen? He shared the Nobel Prize in 2012. So one of his experiments, which was done almost 20 years before the Nobel Prize, Gurdon is a developmental embryologist, biologist. And he wondered what would happen if he removed the nucleus from an intestinal cell of a frog and put that nucleus into a frog egg which had been enucleated. And when he writes about it in his Nobel lecture, there are various ideas that he expresses as to what might happen, but he wasn't convinced that he knew until he did the experiment, and what happened was that a tadpole emerged. And this was one of the first experiments to indicate, this is a long time ago, differential functions of a nuclear nature and otherwise of a cellular nature. Well, now we jump to more recent times with Shinya Yamanaka. And his story is also, I think, very important for all of us. Yamanaka was trained as an orthopedic surgeon. He was very much into martial arts and had many accidents and a few broken bones. And he says, that's what led him into orthopedic surgery. Now, what's interesting and what he told us when he was visiting here the year that he won the Nobel Prize, after being a surgeon and repairing injury, particularly in inheritable diseases of bone, he was curious about what caused the disease in the first place. And that's what led him more into a biological approach. And he then spent about 14 years at the University of California in San Francisco, and also in Osaka, where he got a Ph.D. In molecular biology. He wanted to return to Japan from San Francisco. His wife wanted to go back home with the children, and so he applied for a job at his university, which then was Kyoto. And some wise committee said, "We will accept you, but you have to propose something that's really exciting. Not just a continuation of something that's going on or something that's almost repetitive or prove something that seems to be pretty solid, but really out of the box." And he tells the story about reading of earlier work by the experimental embryologists. And he came up with the idea that maybe it's possible to take a differentiated cell and have it go backwards into an undifferentiated stem cell. And one of his professors convinced him of a motto, which he also told us. He calls it the Volkswagen model. The V stands for vision and the W stands for hard work. And his whole career illustrates that, because these experiments that he performed did not happen overnight. They happened because of diligence. Now, what did they do? He took a differentiated cell and an embryonic stem cell, and they did complete sequencing on both of them and identified various gene candidates that were expressed in the human embryonic stem cells, but not the differentiated one. So, with a group of very diligent graduate students who spent 3 years doing this, and for the first 2.5 years getting nowhere, they were replacing one gene at a time into the undifferentiated, into the differentiated cell to see if there was reversion. It didn't work. They put 2 genes at a time, 3 genes at a time. And then finally, as he tells the story, the student said, "We're doing this backwards. We should put them all in and then subtract." And that's what they did. And they eventually narrowed it down to 4 transcription factors indicated on the slide. And those transcription factors, when introduced to a differentiated cell, cause its reversion back to essentially an embryonic type stem cell, an induced pluripotent cell, iPS. Well, once that happened, this was like a brave new world. And it became an area of intense research and an area of intense hype. People began to think in terms of now regeneration. We can make tissues and replace what's old. We can use tissues to test drugs. We can do all kinds of things, some of which is in the pipeline. But it's been quite a long pipeline, and it hasn't been an easy process. At any rate, Yamanaka shared the Nobel Prize in 2012, and I put here the title of their 2 Nobel lectures, which, if you read anything in the reference material that we post all the time, I would urge you to read these, because they are not only elegant literature, but they tell the story of the human being who actually did the work and why and how it happened, and it's motivating. Now, everybody who uses vision and hard work doesn't come out with a Nobel Prize, and probably more often than not, you have to know when enough is enough and go on to something else. But when it works in a way that is most dramatic, and certainly this discovery of the iPS cell is profoundly changing the way in which we think about biology and also about medicine, and that's the nature of the bridge. So today's participants on the bridge are two NIH experts in this field. The first is Pam Robey, who's a senior investigator here. Pam got her Ph.D. from Catholic University and came to the NIH in 1979 and has progressively worked primarily in diseases and biology of bone, particularly the role of mesenchymal stem cells in bone as leading to different cell types. And because of this work and her, really her expertise, she was appointed as the acting director of the NIH Combined Stem Cell Unit, which she is at the present. She's received many awards and is widely published in the area of her interest. And our second speaker is Kapil Bharti, who is a Stadman investigator in the National Eye Institute. He's the director of the Ocular Stem Cell and Translation Research Unit, which is designed to do translational research involving degenerative eye diseases based upon the use of iPS cells. Dr. Bharti trained in biophysics in India and then received his Ph.D. from the Goethe University at Frankfurt in Germany. He came to the NIH in the NINDS as a postdoctoral fellow, and his major efforts and work have been in the area of transcriptional regulation of pigment cell biology and the eye, and the use of the eye as an organ for therapeutic use of iPS cells. So we're grateful to both of you to be with us, and I guess, Pam, are you going to speak first?
Well, thank you. I really appreciate the invitation to talk to you a little bit about stem cell biology today. I apologize to those of you who are experts in this particular area, but I have to tell you that in my travels around the country and around the world that there is still a great deal of confusion about what different kinds of stem cells are and what they can do and what they cannot do. What I'm going to do is start off with a little bit of stem cell history and definitions. I'm going to talk about the development and types of stem cells, some of their potential uses, and I will use examples of skeletal tissues because that is my particular area of research. And I'm going to talk a little bit about some success stories in terms of using stem cells in clinical applications, and then Kapil will tell you about his very exciting work in terms of retinal pigment epithelial cells. So to start off with, the stem cell hypothesis is not really a new thing. It was put together by a bunch of really astute hematologists and developmental biologists working in the late 1800s and around the turn of the century in the 1900s, in particular Rigaud, Weidenreich, Danchikov, Maximow, and others. I'd like to point out that Danchikov, Vera, was one of the first women professors in all of Europe based on the work that she did in developmental and stem cell biology. And what these people noticed were that during embryonic development and the growth of an organism after birth, tissues are continuously remodeling and regenerating. And the only way that this could happen is if there is a stem cell that is fueling the fire, so to speak. Without having a stem cell, once the cell dies, then the tissue would languish and you would no longer have a functional tissue. But what they didn't really notice at the time and has come along since then is that there really is a hierarchy of tissue turnover and tissue-specific stem and progenitor cells. There are some tissues that are replaced at a very, very high rate, such as blood, skin, and the gastrointestinal tract. There are tissues that have a very slow rate of replenishment, such as bone. There are tissues with limited ability to repair, such as muscle and also the innermost hard surface of the tooth, the dentin. And then there are tissues that were never thought to repair, such as nervous tissue. But all of these tissues do, in fact, have a stem or a progenitor cell that will fuel the fire and replace small amounts of tissue at least. However, it's not one size fits all. Stem cells are a variation on a theme. And that variation is based on the rate of tissue turnover and the specifics of various different tissues that contain stem cells. So if you were to ask a room full of stem cell biologists what the definition of a stem cell is, you would get about 10 different definitions based on their tissue of interest. But there are two properties of stem cells that everyone can agree on. And the first one is that the offspring of a single cell are able to rebuild the functional parenchyma of a tissue. In other words, the working cell type, not the stroma, not the capsular kinds of tissues, but the functional part of the tissue. And that a stem cell has the ability to self-renew. So what exactly is self-renewal? Because this term is abused more frequently probably in stem cell biology than any other term. And that is how a stem cell keeps itself replenished. So it's thought that in postnatal stem cells, that a stem cell undergoes an asymmetric division where one daughter cell is a more committed cell that goes on to transiently amplify and then to differentiate into a post-mitotic functional cell. But then the second daughter cell remains a stem cell. It turns out, though, that it's a little bit more complicated than that. Sometimes a stem cell will undergo a symmetric division, meaning that the stem cell is not being replenished, and you have stem cell loss. And then there are other situations where the stem cell divides and both daughters are stem cells, another form of symmetric division. This may be something that is happening when you have a tumor formed. This is what you have happening when you have ES or iPSC division. And in fact, in a tissue, the stem cell may toggle back and forth between symmetric divisions and asymmetric divisions, depending on the microenvironment. And we truly do not have a good handle on how stem cells in each individual tissue self-renew. It may be many different processes depending on the microenvironment. But the one thing that we do know is that self-renewal does not mean extensive proliferation. And time in, time again, you will hear people say or read in papers that they have a stem cell because those cells proliferate extensively. And that does not make the cell a stem cell. There are other criteria, meaning that they have the ability to differentiate into a functional tissue. The other properties of stem cells are really assumptions that we've given to these cells based on what they do in a culture dish. It's thought that stem cells are clonogenic. As I mentioned, that self-renewal means unlimited proliferation. And it's also been thought that stemness means that the cell type is undifferentiated. But the fact of the matter is that this is not necessarily true for all stem cells, and that we have to find out what the dynamics of each individual stem cell is and not try to lump them all in the same box. So if we start off in development, we have the fertilized egg, which is totipotent. This is what Elizabeth Hay, a very famous developmental biologist who died just a few years back, would call the mother of all stem cells. And it's the mother of all stem cells because not only does it create every cell in the body, it also creates the placental membranes. So this is what is a totipotent cell. Which goes on to divide to form the blastocyst, which has the inner cell mass. The inner cell mass contains pluripotent cells. These cells will form every cell in the body. This is the source of embryonic stem cells, but it's not able to form the placental membranes. And it by itself cannot form an embryo. It needs the extra embryonic membranes to do that. But in terms of what these pluripotent cells can do, if you were to take a mouse ES cell or a pluripotent cell and put it into a blastocyst, the embryo will form. This was done many years ago by many different people. In vitro, they will form virtually every cell type in the body. If you don't allow them to attach, they form what's called an embroid body, which contains tissue types of all different kinds that are found in the body. And if you put it into a mouse, it will form a teratoma, which again has all of the differentiated cell types. So based on these types of assays, we can determine the potency of a given population of cells. As development continues, the embryo undergoes gastrulation to form the 3 germ layers: ectoderm, mesoderm, and endoderm. These cells are multipotent. A single cell from one of these germ layers cannot reform an embryo. They've lost some of their potency, but they have become lineage committed, and they are multipotent stem cells. And then after birth, as I mentioned, we find in many different tissues, if not all tissues, some form of adult stem progenitor cell that is able to refuel formation of tissues and participate in tissue turnover. So again, during development, the specification is not finished at gastrulation. For example, the mesoderm becomes segmented into paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. And then as development continues, even more segmentation occurs. For example, paraxial mesoderm forms the somites, and somites go on to form at least 4 different tissues. So there is continual specification as the embryo is growing. And so as when development is finished, approximately 220 different cell types are formed just from that one single fertilized egg that has undergone this particular process, which is really a remarkable thing when you think about it, that actually embryonic development occurs normally in virtually the vast majority of times. So as Wynn mentioned, we underwent a revolution starting in 1998 when Jamie Thompson first created human embryonic stem cells using the teratoma assay, which is really the gold standard by which you judge pluripotency. He had representatives of all 3 germ layers, gut-like tissue, bone, striated muscle, cartilage, and glomeruli, which is endoderm. And then, as you heard, Yamanaka using 4 factors, OCT, SOX2, KLF4, and c-MYC, also using the teratoma assay, again showed pluripotency in these reprogrammed cells. And then Jamie Thompson, using different factors, also derived human pluripotent stem cells using human somatic cells. And I'd like to highlight that in addition to the teratoma formation, the surface expression of these markers, SSEA3 and 4, TRA160 and TRA181, and also OCT4, NANOG, SOX2, and LIN28 are markers that are typically used to determine the pluripotency of a population that has been generated and are quite useful. So after the first discovery of the reprogramming using viral transduction, a number of other different methods have been developed for creating pluripotent stem cells, including viral transduction with many different types of vectors and viruses, plasmids, minicircles, protein transduction, RNA transduction. And I think right now, basically the 2 that seem to be working the best, according to Barbara Mallon in the stem cell characterization facility, are using sendivirus with the 4 factors and RNA transfection. But there are real issues in terms of reprogramming. Not everything that you generate in the dish is actually pluripotent. So there's the quality of reprogramming that has to be evaluated. It turns out that there are different substates of pluripotent cells that give them a particular preference for one embryonic germ layer than another. There are times where you have not completely wiped out the memory of the cell, so it remembers a little bit about where it came from. And then there was the issue of naive versus primed. Naive are cells that are a little bit more primordial and better match cells in the mouse embryo. Primed appear to be ready to go into one particular germ layer or another. And there are a lot of papers about this from these 2 workers. And then there's also what I call direct reprogramming. And this is exemplified by this paper from Noriyuki Sumaki in Japan, where he took skin fibroblasts and transfected them with c-Myc, KLF4, and SOX9. SOX9 is the master gene for cartilage. And then he developed what he called these icon cells, which if you put them into a pellet culture, they form beautiful cartilage. And this is in comparison to the parental skin fibroblasts, and also in comparison to other cells that form cartilage. The cartilage looks really good. The pros of this approach is that there's less risk of teratoma formation, and you can also sort these cells for markers of chondrocytes and purify them. The cons is that, unfortunately, many of these cells formed chondromas, which are actually worse than teratomas. They're very nasty tumors. And then the other problem is that depending on the cell type, you have very, very limited expansion after you've directed them using these transcription factors. And so generating enough cells could be a problem with this type of message. So in terms of the various different types of stem cells, I've kind of highlighted how they come about during development and how we create them in a dish. What I would like to highlight now is that we need to know about all kinds of stem cells if we're going to use them successfully in tissue engineering and regeneration. For example, we need to know from embryonic stem cells and iPS cells what gives them the capacity to proliferate continuously, and what makes them pluripotent. What are the factors? And I think that we only are scratching the surface on our knowledge. In terms of fetal stem cells, we know that they can proliferate extensively. They're multipotent, but they're committed to a particular lineage. And so what are those factors? And then finally, there are the postnatal stem cells. We know that they have very controlled and predictable differentiation. So what better cell type than a postnatal cell to educate an embryonic or a pluripotent cell on how to differentiate? And what better cell type than an embryonic or an iPS cell to tell a postnatal cell how they can proliferate more extensively? So one cell type is not going to get us to where we want to be. We want to be looking at all of these different kinds of stem cells. So what can we use stem cells for? Well, we can use them for development. For example, we can determine how a complex organism can emanate from a single fertilized egg. We can follow the stem cells as they divide and become more specified. We can use them to identify the signals and the mechanisms that determine stem cell self-renewal versus differentiation. This is really important in terms of tissue homeostasis. And we also can use them to determine how they make their lineage commitment choices. So for example, in bone biology, cells can become either bone or cartilage. How do they decide which way they're going to go? And we can use iPS cells to try and understand these. And so Kevin Egan has actually put together this roadmap on how you can get pluripotent cells to differentiate into all the cells in the body, You'll hear about this retinal pigmented epithelium from Kapil. In terms of the osteogenic and chondrogenic differentiation, I have some misgivings about his map here, but it's a start. You have to start somewhere. But this is a work in progress, and this is really the goal, to be able to get these cells to not only differentiate into a specific cell type, but also to get them to differentiate into a mature cell type. This has been a real problem in the field, that we can get hematopoietic-like cells, but they're not mature. You can get muscle-like cells, but they're not mature. Does it matter? If we put them in the body, will the body tell them how to mature? Or do we need to tell them how to mature in the dish? In terms of osteogenic and chondrogenic differentiation, we started off treating the cells just like they were bona fide osteogenic precursor cells, and we were able to get small amounts of bone and also small amounts of cartilage. One line formed both bone and cartilage, but this was not robust. We were not getting the amount of bone and cartilage that we would get from bona fide chondrocytes and bone. So the next approach was to follow a more developmental line, and that's being done by Fahad Kidway in my lab. Basically, it turns out, we knew for a long time, but others didn't know, that there are at least three different sources of bone. There's the neural crest, which gives rise to facial bone and cartilage. There's the dorsal part of the somite, the sclerotome, which gives rise to the axial skeleton. And then there's the somatic lateral plate mesoderm, which gives rise to the appendicular skeleton. So three different embryonic sources. And it makes a difference in how you treat them to get these different embryonic sources. And so this is kind of the roadmap that we're using at the moment to try and get them to differentiate into bone and cartilage from the three different germ layers, because it's our hunch that it matters if the cartilage or bone is neural crest in origin versus lateral plate or paraxial. There are some clinical indications that it really does matter where it comes from. You can use stem cells to study disease. So, for example, it's very difficult often to get sufficient cells that are from a patient to study them in detail. If we can create stem cells with a disease-causing mutation or engineer them, that offers a viable alternative to studying the pathogenetic mechanisms. And we can also use them for in vivo transplantation into humanized or immunosuppressed mice to develop a diseased tissue in order to try and come up with a mechanism. And in fact, we've done this. For example, using skeletal stem cells, which are the cells that I work on, we hypothesize that because these cells are very important in skeletal homeostasis, that any genetic mutation or any change in the microenvironment that changes their activity will result in a disease. And our example was fibrous dysplasia of bone. And basically what we did was we isolated skeletal stem cells from normal bone and from fibrous dysplastic bone. You can see this is very abnormal. There's no marrow, woven bone, very weak woven bone. If we take those cells and we put them onto a scaffold and transplant them into a mouse, the normal skeletal stem cells make a normal bone ossicle, and the ones from the abnormal marrow make what we call an FD ossicle. So it's recapitulated the formation of an abnormal bone in a small animal. And so this is a model that we can use for studying the pathogenic mechanisms and also to come up with different ways of treating these cells to reduce the effect of the mutation. There have been a number of articles written about using pluripotent stem cells in terms of modeling disease. And this is one of them, talking about using the cells for 2D models, 3D models, and also 4D by adding in the element of time, watching how a tissue evolves, whether it be in an organoid or on a plate. Tissue on a chip is being engineered in many different labs around the world. And also in terms of RPE, there are studies where there was a mutation in mitochondrial DNA that was used to study the dysfunctional RPE, where you had very abnormal swollen mitochondria and abnormal melanosomes. And so these cells can be very useful in terms of coming up with models of disease. And in order to do this, we can start off with normal cells and use the emerging technologies for genome editing, where we can modulate, create, or correct mutation. And there are a number of different methods using pre-locks for modulating the level of a mutant protein. CRISPR, TALEN, and zinc finger have all been used to either create mutations or to correct mutations in iPS cells. We can use stem cells to replace damaged tissues. So this offers a way to correct a segmental defect that would never heal on its own. This is being done with a limited number of postnatal stem cells, such as hematopoietic stem cells, which are being used in bone marrow transplantation for hematological disorders. And we can really look at them as a very exciting opportunity, but there are a lot of hurdles that we have to jump over before iPS cells will be ready for prime time. And I think Kapil's project is one of the prime examples that looks very, very promising. So in terms of other stem cell-based therapies, for example, there's Parkinson's disease, which initially was treated with fetal neural tissue. Those studies did not work very well, but it's now actually being revisited because they learned a lot of lessons about how to transplant these cells. Whether fetal tissue will really be useful for treating Parkinson's, we don't know, but this review article talks about what they learned from the early trials and what they're changing now in the later trials. And it's a European consortium that is now doing this. In terms of bone regeneration, we know that we can take bone marrow stromal cells and skeletal stem cells. We can attach them to a scaffold and put them into a segmental injury. We can also correct their gene mutations. We can also inject them directly into an injury with a injectable carrier and regenerate bone. And this has actually been done in a few humans. 3 different patients have been treated with these cells along with hydroxyapatite. And you can see that there is a little bit of bone regeneration along the edge of the scaffold that was used in this case. And we are working very much on trying to reconstruct craniofacial defects such as calvarial defects and jaw defects using this technology. And here you can see that new bone is formed by the cells, and they also support the formation of marrow. And we're working on developing a protocol for this in the Dental Institute. I hope that someday before I die that I will actually get to do this protocol. There's a lot of excitement in terms of treating patients with skin diseases. This is a terrible disease called epidermolysis bullosa. It's caused by mutations in one of the laminin genes, LAM beta 3. This marks the areas where there was either blistering or complete denuding of the skin from this boy with this disease. And you can see that he's completely missing LAM 332, which is laminin 5. They took his epidermal cells, stem cells, they transfected with the defective gene, expanded, and they have pretty much saved this patient. He is completely covered with normal skin at this point, and it really has a big breakthrough. You'll hear from Kapil about regeneration of RPE. And also we can use stem cells for testing new medical treatments. This is one of the things that they are doing out at NCATS, trying to develop ways of using various different differentiated progeny of stem cells, using their libraries and their robotics to screen for new drugs to treat various different diseases. So I just want to briefly mention the resources that we have here at NIH to do this kind of work. The first is the NIH Stem Cell Unit that Barbara Mallon is the technical director, and she basically has done a huge amount of work in optimizing the growth and the generation of iPS cells and establishing standards. She has a database that's on the NIH website where you can plug in the data from your stem cell to compare it to the iPS cells or human embryonic cells, and she provides a tremendous amount of support to the intramural investigators. And this is an example here of deriving stem cells from either skin fibroblasts, CD34 positive cells, or from my favorite cell, bone marrow stromal cells, and using either embryonic fibroblasts as a feeder or matrigel, and also doing monolayer cultures. And she and Kevin Chen and Kai-Yun Park have worked out all of these methods. Then the other thing that you have to think about in terms of differentiation is whether or not you're going to use embroid bodies, which contain all of the differentiated cell types, or monolayer cultures to do your differentiation. The pros are for embroid bodies is that you can take your embroid body, digest it with an enzyme, and then do FACS to isolate the cells with the cell surface character that you want to isolate. But the con is that the embroid bodies are extremely variable, and then the problem is that the number of cells that you can get and their proliferative capacity may be quite reduced. The pros for the monolayer is that you can generate large numbers of cells. It's very easy to scale up. But the con is that unless you really know what your developmental sequence is, you can end up with a really mixed population of cells that really will not perform the way that you want them to. The other resource is the Department of Transfusion Medicine, the cell processing section, headed up by David Stronczyk, and their mission is to collect, manufacture, and store cellular therapies. They do a lot of donor evaluation, laboratory testing, and they have a lot of medical consults with people that are using CAR T cells, for example, to treat various different types of blood disorders. As you can imagine, and Kapil will talk about this, there are a huge number of issues that you have to deal with before the FDA will approve your cell product for use in humans. Identity and purity are really the main ones, and efficacy, stability, sterility, these are all things that Kapil will talk to you about. So this is my last slide. Basically, it lists the pros and cons of different kinds of stem cells. Basically, it just depends on what your particular application is. In terms of the purity of the population and what you're going to use it for, you really need to kind of tailor your cell product to the application. In terms of human ES cells, they are the purest population because they have not been genetically manipulated, but there are ethical issues and there are problems with teratomas. iPS cells don't have the ethical issues, but we really don't know how to differentiate them very well. Fetal cells have ethical issues. We don't have large numbers of these cells. Postnatal cells are very good at differentiating into their specific tissue, but we can't get enough of them. And you can go on, and you can end up with a really long list of pros and cons. And so it's a matter of really trying to tailor it to what your application is. So at this point, I just want to acknowledge many people here, Alexander Friedenstein and Paulo Bianco, who really studied the skeletal stem cell and what it could do in the bone marrow microenvironment. My former fellows, Mara Ramanucci, Mahesh Mankani, Paul Krebsbach, and Matt Phillips, who worked on these various different cells. This is the current group here who are working on single cell analysis of various stem cells. Here's the stem cell characterization facility with Barbara, Kevin, and Kai-Yun. David with the cell processing section, and Mike, who's in the crowd, who is my partner in crime in terms of fibrous dysplasia of bone and some of the work that we did early in that game. So at this point, I'll stop and ask if you have any questions. Feel free to contact me. This is my email address. I'm more than happy to share my slides with you if they would be useful to you or helpful to you in that any way, shape, or form.
Thank you very much. So we have time for some questions. Please use a microphone because people listening want to hear the questions as well.
And the speaker wants to hear too because she doesn't hear very well.
Hi. I'm interested in the chemical modulators that are being used, and you touched on it with NCATs, but I'm wondering, is this a rational strategy, or is it just screening randomly through libraries? Because you mentioned, and Wynn mentioned the 4 genes that can perform this transformation.
Mm-hmm.
Are there other genes that can be modulated chemically to produce the same type of transformation?
Well, I mean, the fact that Jamie Thompson and Yamanaka came up with 4 different combinations means that there is redundancy. And so we just actually had Elias Singach, that's how you say it, give a seminar in the NIDCR. And he basically has been doing some screening with large libraries, but he's also been doing screening with specific pathways. So he's hedging his bets.
So I'm one of those people who learned today that there are different origins of bones in the body. Thank you. So using that knowledge, the question is the bone marrow derived stem cells to treat, let's say, femur versus what you're trying to do for the facial bones. You have to use different type of bone marrow stem cells. So can you use heterologous bone stem cells?
There's anecdotal and a few papers from the dental field saying that if you take iliac crest and you pack it into the jaw, it disappears. And so Fahad is really going to try and test that hypothesis by taking cells from that have gone through lateral plate and putting them into the jaw and vice versa to see what the interactions are. Perhaps we will have to teach iliac crest how to be more like neural crest.
So I wonder, one of the problems is with the iPS cells, many take a long time, weeks, months, before the conversion takes place. And so there aren't a large number of cells, but the phenomena has taken place. So is the rate of conversion related in any way to the biological turnover of those cells in the intact organism?
Intuitively, that's what people have thought, but I'm not sure that there's any data that supports that. Certainly, their memory, whether they were very differentiated or a progenitor, could also be impacting on how fast. But if you think about it, the epigenetic landscape has to be changed so dramatically, and I think that just takes time.
So you have a bone growing in a petri dish. Are there factors known that determine the final shape of that bone, ossicle or femur, without having it in place where they?
Yeah, mechanical forces are everything, and in our subcutaneous ossicle, they literally look like little pancakes, and it's because of the forces of the muscle on the implant. In terms of development, boy, that's like really the $60,000 question, how you get the shape. And the tooth is even more amazing when you think about all the cusps and the roots and things like that. What dictates the shape is really, really not known.
I wonder, in one of your slides, by the way, all of these images are on the website for this and any of the previous lectures. So if you want to contemplate some of the detailed information, you can go there and see it. So you, in passing, talked about the putative cancer stem cells. and you sort of put it on the list as one of the somewhere in between. But it seems to me there's a huge debate as to whether these are truly stem cells or just cells that are progenitors in a sequence. Did you want to comment?
Yeah, I think I come down on the side that these are just progenitors in a sequence that for some reason can't go on to the next step of differentiation. And, you know, in terms of what the cancer initiating cell, I'm more in favor of that term than a cancer stem cell. It's what initiates and propagates. And where it comes up is a big question. For example, in osteosarcoma, my gut feeling is that it's a bone marrow stromal cell that starts to proliferate, has its various different mutations. There are a number that are known, and it just cannot go on to the next step. It hasn't gone back to the most, as far as I can see, it has not gone back to the most primordial cell in the lineage.
Is there a role for stem cell therapy in fibrous dysplasia?
Well, it's a somatic mosaic disease, so yeah, we could regenerate bone from their normal cells. Michael is developing small molecules to try and knock down the activity of the mutated gene. I think that there are other diseases that are probably, the fact that we can get normal cells from these patients is a good thing. There are other diseases that where we would have to correct the mutation before we could use stem cells.
Okay, well, thank you. There'll be time for more questions, and so now we'll go on to Dr. Bharti.
I want to start by thanking Wynn for this invitation to talk about our work, and actually also want to thank Pam for giving such a broad overview on stem cells. So that gives me the opportunity to directly focus on really the work that we're doing in the lab in terms of developing cell therapy using iPS cells. So the disease I'm going to focus on today for this stem cell therapy is age-related macular degeneration. Just to give you a brief overview of what this disease entails, on the left side you see, or actually on my right side you see this how most of us see is a normal vision of a person, and on the other side you see how a person with age-related macular degeneration (AMD) sees. In the central part of their vision, they're going to be completely blind wherever they would look. And this is a disease that affects mostly elderly people. There's several million people worldwide that are affected by this disease. And currently, for one specific form of this disease called the dry form, which affects almost 90% of the people with AMD, there's no treatment. So this is a big unmet medical needs in the field. So if we kind of look into the back of the eye and try and understand how this disease happens, the next couple of slides that I have shows a schematic of how we currently best understand the kind of pathogenesis of this disease. So this would be a healthy eye. On top, you see the retinal neurons, which are connected to the photoreceptors, really the main cell type of the retina, which perceives light and sends signals to the brain so we could all see. And right next to photoreceptors is this layer of cells called the RPE, the retinal pigment epithelium, that is the topic of discussion for today's talk. And what you would notice is a couple of interesting features of this cell type, which is, first of all, it has this very strategic location. It's present between the photoreceptors and the blood supply. And the reason for that is that these cells are highly polarized, and they have tight junctions between neighboring cells that form the outer blood-retina barrier. So what that means is that blood is not in direct contact with the photoreceptors. All the metabolites that have to go to the retina have to pass through the RPE cells, and all the nutrients have to pass through the RPE cells, and all the metabolites have to pass back again from RPE into the choroid blood supply. And the other thing you notice is they have these hair-like structures, what we call as apical processes of these cells, and they sit on this proteinaceous Brooks membrane. And these hair-like structures are really what we call as the business end of RPE cells, and they interact with photoreceptor outer segments and help perform all the functions RPE cells do throughout their life to maintain health and integrity of these photoreceptors. So they replenish visual pigment for photoreceptors. They eat our worn out outer segments of photoreceptors. So all of this tissue is really there to help the photoreceptors survive. So obviously, in a disease like AMD, where RPE cells die off, photoreceptors start to die. And that's the reason where people really go blind. And so this is what would be kind of the central part of a person with AMD. You can see the photoreceptors are gone, the RPE cells are dead, and even the capillaries are starting to thin out. But if you move away from this area, the central area where those people are blind, towards the healthy part of the eye, there's this area called the transition zone. It's called the transition zone because if you notice here, the RPE cells are missing, but the photoreceptors are kind of still hanging in there. And it's thought that these photoreceptors are dysfunctional. They are likely destined to die because they don't have the RPE support. But what surgeons have tried for a while and have thought for a while, that if you were to bring a transplant of RPE in this transition zone, you could protect these overlying photoreceptors from dying. And this has been tested surgically. So if you think about a whole eye and you have damage in the center part of the eye, they would cut out a piece of RPE from the periphery of the same eye, translocate that into the center, into this atrophy area in this transition zone, and leave the transplant there. And in small number of cases where the surgeries were successful, they saw that patients' photoreceptors were surviving for several years to come, and patients' vision was rescued. At least it was stabilized for years to come. And these are some of the papers that have previously demonstrated that. And for me, this is a critical proof of principle experiment that if you were to make an autologous RPE patch and from AMD patients and deliver it to the right place, you could actually save their vision from getting worse. And that is really the driving principle for us to make RPE patch for AMD patients. So we start with the patients on cells, as Pam explained, the iPS cell technology, reprogram their somatic cells into iPS cells, differentiate those cells into an RPE patch, RPE cells make a patch and deliver it back to the patients. So this slide shows you a small video of how the patch looks like. It's a 4 by 2 millimeter patch that covers roughly half of the macula, half of the center of the eye. And what you see in here is RPE cells are on top. You can see these pigmented cells. And when we flip the patch, you will see they are sitting on a scaffold, on a biodegradable scaffold, which is color white here. And the idea is that the cells would make their own ECM, and as they integrate, the scaffold degrades, and cells continue to sit on their ECM and integrate in the back of the eye. So what I'm going to do today is kind of go over the whole process of how we actually make this patch for transplantation, the 3 parts of an IND application that we need to start a phase 1 clinical trial with these cells is how do we manufacture iPS cells, how do we manufacture RPE patch from AMD patients, what is called the CMC part of the IND application, chemistry, manufacturing, and controls, and then how do we test these patch in preclinical animal models to demonstrate that the cells are safe, they're not tumorigenic, they're not toxic, and they do have some efficacy in these animal models. And then how do we demonstrate that the tool and the scaffold that we have made to deliver this patch is also biocompatible, and of course, eventually we'll have a clinical protocol how we deliver it to the patients. So just a brief overview of the manufacturing process or of the developmental process of how we make RPE cells. So this slide shows you from iPS cells all the way to mature RPE. The process takes about 10 weeks. It's a fairly long process, but it's well optimized. But the important point to remember here or to realize here is that iPS cells are coaxed into various developmental stages, kind of that big overview that you saw in the previous talk. The cells go from a neuroectoderm stage through RPE progenitors all the way to mature RPE. And we do that by manipulating these growth factors at various time points. And this is just really reproducing developmental biology of RPE cells in a dish. All the work that we have learned from over decades in various animal models, how we make RPE cells, we try to reproduce that on iPS cells in a dish. And interestingly, in the case of RPE cells, we actually can get functionally mature RPE cells. It's one of those rare cell types. We just recently published that we can make it into completely mature RPE cell, and that becomes relevant when you're transplanting it into older people. You want to transplant likely mature RPE cells that would start functioning right away. So the bottom panel of this slide shows you that indeed we can get pure cells. This is a dish full of pigmented RPE cells, and by flow cytometry we can show that these cells are positive for a number of different RPE markers, and this also demonstrates that the cells are fairly homogeneous in terms of the expression of RPE marker. An important point you also we have to consider is that we are trying to develop an autologous therapy. So if you have to make cells from everyone in this room, the process has to be highly reproducible. And here we have tested this process now over 34 different donor iPS cell lines from healthy individuals, people with different types of diseases, clinical grade cells, research grade cells. And pretty much in every single case, we were successful in making RP cells and mature RP cells from iPS cells. So that's an important point going forward, that this process is really reproducible in making RPE cells. Once we have a relevant cell type, we put them on this scaffolds, the one that I showed, biodegradable scaffold. These were designed such that, as you can see, these are electrospun nanofibers, about 300 to 400 nanometer diameter, nanofibers of PLGA, polylactic glycolic acid, that are fused so that the cells would form a monolayer on top. And then yet the scaffold will have small pores to allow nutrient flow, and pores where cells can deposit their own ECM so they can form a Brooks membrane. And the next slide nicely illustrates that the cells do end up forming a nice membrane structure. So this is a scanning EM of RP cells grown on a PLGA scaffold. In this case, it's pseudo-colored to show you the scaffold and the ECM in blue. And since you're looking at this structure from a side angle, you see a nuclei of 3 cells. There's one cell, another one, and another cell. But you see this in green apical structure of all the cells on the monolayer. And these were, remember, the main business end of these cells. And I hope you can appreciate that pretty much all the cells have extensive apical structures, apical processes, and they're pretty homogeneous throughout the tissue, suggesting that really we can make polarized cells using iPS cells. And not only we show that by structural assays, we have extensively functionally characterized these patches, and this is one example of a kind of function these cells can perform. So these are epithelial cells, and I mentioned that they have tight junctions between neighboring cells that form the outer blood-retinal barrier, and because of that tight junctions, they have what is called electrical resistance, transepithelial resistance. So what I mean by that is, if you put an electrode on top, one on bottom, you pass current, cells would show a resistance to the flow of current. And that resistance is an indication of maturity of cells. So we can measure that resistance as, in this case, several hundred ohms per square centimeter, showing us that these cells are mature. And at the same time, because of the tight junctions, the two membranes, apical membrane and the basal membranes, are isolated. They have different receptors and channels on two sides that can be measured as a different membrane potential of the apical and basal membranes. Again, a feature of polarized cells. And then we can perform perturbations like changing potassium on the apical side or ATP on the apical side, physiological changes that happen in the eye, and these cells respond by hyperpolarizing or depolarizing, and they show responses pretty much similar to what native RP cells do. So to demonstrate the proof that these cells are not only structurally mature, these cells are also functionally mature, made from iPS cells on a scaffold. So then we spend quite some time really optimizing and validating how do we make these cells under GMP grade settings, because as you may know well, if you have to transplant any cell therapy product in patients, it has to be done under the current good manufacturing practices as FDA requires. So you're talking about the process is long. Yes, it is long. It takes about 6 months to make, start with the patient on day 0 and to have a final product ready for the patient. But so we go from, in this process, in this case, isolating CD34 positive cells from patients, making, reprogramming them into iPS cells that we can expand to a small, what we call a working bank at passage 10 of iPS cells. This itself takes about roughly 3 months. And then it takes about another 3 months from that iPS cell bank to making a mature RP product. Similar to what I just showed you, we go through the progenitor stage, seeding cells onto the scaffold, finally getting a mature product that we can release for transplantation. Now, if the process is so long, we have to have quality controls during the manufacturing process at various steps to see if it's going in the right direction. So, for instance, we characterize our iPS cell bank thoroughly for sterility, quality of iPS cells, and then when we start differentiating them into RPE, again, we make sure that the differentiation is going in the right direction, and finally validate the product before we transplant. And then at the same time, we have put in place several steps where we can cryopreserve our intermediate products, so that in case the process fails at any of the steps, we can go back to either the patient starting CD34 positive cells or one of our iPSC banks or the progenitor RPE bank to restart the process and not have to go through the whole 6 months of manufacturing again. So this process is now being done at the DTM, Department of Transfusion Medicine, Pam mentioned in Dave Sonchek's unit, has been tested on a couple of different healthy donors and AMD donors, and it works even reproducibly under GMP conditions. So again, one of the critical parameters is when you're making autologous cell therapy that you want to have a way of validating what is the transplantable tissue, because there's going to be some heterogeneity in tissues made from different people. So to really understand how much is the heterogeneity and how can we actually say that this RP patch is transplantable, whereas this one may not be. So to do that, we made cells from 3 different AMD patients, and we made iPS cells from 3 clones per patient. So we had 9 clones. All of those differentiated into an RP patch under GMP grade conditions. And we characterized those patches extensively for these different assays that I have listed here, including, so the goal was to look for if there's genetic differences between different people affect the quality of the end product, if there are technical differences in different assays that affect the quality of the end product, and what are the different assays we can actually use to characterize this end product. So I'm gonna, so there are 2 assays that actually really stand out. One of them, transepithelial resistance, that I just talked about, measuring the electrical resistance of the monolayer. That really for us is a key parameter to tell how mature the cells are. And the second that I want to briefly mention is the quantitative shape metrics. So what we do in this case is we use neural networks. So we have developed a neural network that can just look at the image of RPE cells and tell us how good the cells are. So what we did is to develop that network, we wrote the code, and we fed it with images of RPE cells as the cells were maturing, and we fed these functional parameters, TER, transepithelial resistance. And then after training the network, we asked the network to predict function of RP cells, which says, can you now look at a picture of cells and tell us, are these cells functional or not? And this is our really one of the preliminary results we have, because this is just using one training set so far. But the assay the outcome looks really interesting. So what you have on this graph on the y-axis is the predicted TER, the transepithelial resistance that the network predicted. And on the x-axis, we have the measured TER, so that we experimentally measured from the patch. So what we did is we trained the network on 3 different types of RP cells, cells that were fully matured, RP cells that were fully matured, RP cells that were partially matured here in green, and then RP cells that were kept in a fetal-like state. So first thing that you notice is that the prediction of network is almost as good as our measurement that we performed experimentally. You can see the 2 of them are quite linear in the outcome. There's only one mistake the network did. Other than that, they're both very similar in their outcome. And the second more important thing is that the network is able to, just by looking at RP images, can tell mature cells from partially mature cells from fetal-like cells. And this is our threshold that we have kept based on experimental results of about 400 ohms per square centimeter. Above that, we call these cells transplantable. So we're very excited that we think that this machine learning based approach can, because it's so easy to implement under GMP grade settings, can actually tell us as we move forward towards large scale manufacturing a good patch from a bad RP patch that can be transplanted or not. So now in the second part of my talk, I want to move away from manufacturing to now testing these cells in preclinical animal models. So the first test is, once we transplant these cells, once we transplant an RP patch, does it form tumor or teratoma in animals? And these studies are done in immunocompromised animals, because you want your cells, because these are xeno settings, you want your cells to survive long enough that they have the possibility of forming a tumor or not. So here's so-called the positive control, where we are transplanting pure iPS cells in the subretinal space of a rat eye, of an immunocompromised rat eye. This is the lens, this is the retina of the rat eye. And what you can immediately notice is a big, massive teratoma that these cells form, which is what they're supposed to do. iPS cells, when left under uncontrolled conditions, can lead to this teratoma formation. But in contrast to pure iPS cells, when we inject pure iPS-derived RPE cells in the same rats, we do not see any tumor or teratoma in any of these rats. Here's two examples. Most of the times, these cells just sit in the subretinal space under the retina, then they don't do anything. And because here we're injecting cells in suspension, this red is a marker for one of the iPSC protein PML17, and it's human specific, so we can only see human cells in this case. And here on this side, you'll see a marker for STEM121, which is a human specific antigen. Every once in a while, we see suspension cells integrate into the rat eye. We don't know why, we don't know how, but a very small number of cells would integrate in the rat eye, but they don't lead to ever any tumor or teratoma formation. But interestingly, when we transplant RPE patch on that biodegradable scaffold, on that PLJ scaffold, the entire scaffold integrated into the back of the eye, into the back of the rat eye, exactly as we had hoped for. In this case, what you see in red is STEM121, again, the human-specific antigen. We transplanted a 0.5 millimeter diameter patch in a rat eye. And if you measure the scale bar from one end to the other end, it's exactly 0.5 millimeter. This is 10 weeks post-transplantation, so the scaffold has already completely degraded, and the cells use their ECM to integrate, human cells integrated into the rat eye. And you see, here's the rat eye because they're not positive for this stain, and right here the rat eye ends, and the human rat RP ends, and the human RP begins. So this was really what we had hoped for, that transplanting these cells on a patch would increase their integration, would increase their survival in the eye, and that's exactly what we saw. So this result was really exciting. Now, both the suspension result and the patch result led us to perform a massive GLP grade preclinical study at a vendor, in which we have tested a 0.2 millimeter diameter, 0.5 millimeter, 0.2 millimeter square area, 0.5 millimeter diameter RP patch, and 100,000 cells in suspension for 4 different time points. So 2 weeks, 13 weeks, 26 weeks, and 9 months. Animals were sacrificed at these 4 time points, and we looked for whether these cells were toxic, or they were distributing to any other organs, or they were tumorigenic. And I'm happy to report that after performing this on 450 rats, we did not find any signs of any of these toxic events from any of these cells. So these cells are, at least under these immunosuppressed xenograde settings, are completely safe in rats. So then we went on to test whether these cells or this patch has any efficacy, at least in an animal model setting. And so in this case, because we wanted to test our entire patch, a 4 by 2 millimeter patch that we want to transplant in patients, we decided to use pigs as an animal model. And the other advantage of using pigs as an animal model was that pig eye size and eye anatomy is very similar to human eye size and eye anatomy. So we could also optimize the tool, the delivery tool that we wanted to use for transplantation, and the surgical procedure, to how do we deliver it under the retina. But the problem with pigs is that there's no genetic model that looks similar to what happens in dry AMD in patients. So we created an injury model in pigs. What we did is we used micropulse laser to laser ablate RP cells in the back of a pig eye. And the reason micropulse laser specifically knocks out RP cells is that we use a specific wavelength of laser, 532 nanometer, that is absorbed by the pigment of RP cells, because these cells are pigmented, and as over time they just heat up and then burst and die off. So that, and as a result, photoreceptors start to die. So it's a very similar situation to what happens in AMD patients, but we induced it by injuring RPE cells. The bottom panel here shows you some of the results of this laser model by infrared imaging, and that's the other advantage of working with the eye, is that we can do so much analysis by just imaging the eye non-invasively. We can see everything in the back of the eye. So in this case, this is an infrared image of the back of a pig eye where the RP has been laser damaged. And as you can see, because the pigmented cells are gone in this 7 by 4 millimeter area, the infrared light just shines through. Normally, RP cells would absorb the infrared light, and it would not shine through. So what we call this as kind of a window defect. RP cells are gone in this area. And you can also prove that by just performing histology, where you can see RP monolayer is gone. Here's some clumped up RP cells. And as a result, the photoreceptors are dying because the RP support is missing. Now, the idea is that we transplant in this area human RPE patch and ask it to protect overlying pig photoreceptors. So we're asking human RPE patch to transplant in a transplanted pig to protect overlying photoreceptors. So first of all, to deliver the patch, we need to develop a tool. So the tool that we developed has a curvature that fits the back of the eye. It takes the transplant. This is the size of a transplant. It's like a bullet-shaped shape, and it goes inside the tool only in one orientation. So the surgeon would not be delivering it upside down. And then it is connected via a fluidic system through a foot paddle, so the surgeon can control the delivery of the patch back and forth easily just with one hand holding the tool and deliver the patch in the back of the eye. So in the next slide, I'm going to show you a short video how actually we transplant this patch in the pig eye. So as you can see, the eye has already been prepped with 4 ports. The surgeon is making one of the port bigger so they can put the transplant tool inside. And in the next shot, you will see the patch. Here's the tip of the tool. This is a patch. You can see pigmented cells on top. Here's a cut. Here's a retina. The cut in the retina. Retina has been lifted here. And surgeon is delivering the patch right under the retina. And then he'll flatten the retina on top so the patch stays right in that place. And again, by imaging, we can see the patch once it's transplanted. This is an imaging technique we use, what is called OCT, optical coherence tomography, where we can see from the unfast view, here's a patch. You can see this bullet shape of the patch. This was a cut in the retina through which the patch was delivered. And now if we see the side view right here, you can see this is, I've outlined the patch here. You can see this would be the patch right here. And actually, the bright signal on top of the patch shows you that it has cells on top. So now we see it's sitting at the right place. There's a retina on top of the patch, flattened on top of the patch. And then the next step for us is it's structurally at the right place, but is it functioning? So to do that, we use another technique called multifocal ERG. We know the retina has neurons, so it has electrical potential. It responds to light. So if we shine light to the retina, we can measure electrical signals from the retina. But in this case, we use a technique in which we shine multiple different light pulses. Depending on where the light hits, we get response only from a very small area of the retina, and this can be as small as 0.5 millimeter to 1 millimeter area of the retina. And that gives us the advantage that we can, from the same eye, measure signal from a healthy area. We get this nice and big robust response. And then when we shine the light on to a laser injured part, which would be somewhere right here, where we lasered the eye but did not deliver the patch, we get almost no response because the retina is almost dead in this area. But if we shine light onto this part where we deliver the patch, we laser the retina and then deliver the patch, we get a response that is in between, suggesting that human patch is able to rescue the pig retina from dying, and we can measure that electrically. We can also check that by histology. If we do immunostaining for the back of the eye, and this is now a pig eye after the animal was euthanized, we can see human RPE patch, just like in rats, it's integrated into the back of the pig eye. In this case, red is a stain for a human of an RPE marker that stains both the pig RPE and the human RPE, and the green is a stain that only stains human cells, the STEM 121. So you can see this would be human RPE, and would end right here, and the pig RPE starts right here. And you can see the human RPE actually almost starts interacting with the pig RPE. We can also show that the retina is being rescued on top of the patch. This is just another histology section from the same eye. Area that was lasered but no transplant was delivered, compared to area that was lasered but a transplant was delivered, and this is the transplant highlighted in yellow here. Notice the thickness of nuclei. So nuclei are stained here in blue, and these are the ONL is the outer nuclear layer of photoreceptors. So these are nuclei of photoreceptors. Notice the thickness of photoreceptors on above the transplant versus away from the transplant. It's almost double the thickness, suggesting that the human transplant has rescued the photoreceptors, the pig photoreceptors, from dying, and we can at least retain the entire photoreceptor nuclear layer in this case. So this work that I showed you has now led to a preclinical study in pigs, where we have transplanted now over 30 pigs where we tested just the scaffolds, sham surgery, or different components of our patch to demonstrate that the patch is safe and is efficacious. So we're about to finish this study. So just to summarize all of this, what I've shown you is that we have developed a clinical grade protocol to make iPS RPE patch, and we demonstrated the quality of this patch using 3 clones for patients by performing in vitro GLP validation studies and the rat safety studies. I didn't have time to go into rat efficacy studies that we performed, but I showed you some of the data from the pigs efficacy studies. So altogether, this work is being put together for an IND submission, hopefully in the next couple of months, and we hope to start the phase 1 clinical trial perhaps later this year. What I want to show now in the next few minutes is what are we doing next with the stem cells? Where are we taking this whole technology? Remember, we talked about how does disease happen? Disease happens, we think that RP cells die off, and as a result, capillaries become thin and photoreceptors start to die on top of the RP. So what we want to do is we want to really recapitulate the entire disease in a dish. Not just the RPE, we want to recapitulate the capillaries and the photoreceptors to see to really understand how does disease initiate and it progress to such an advanced stage. So to do that, I've already shown you that we can make RPE patch, but now we want to add the capillaries to that RPE patch. So for that, first thing we need to do is make the endothelial cells of the capillaries. So we use a process, again, very similar to what we use for making RPE cells, make mature endothelial cells from iPS cells using a published protocol from Christine Mummery's lab. Works, again, very reproducibly. We have done it over at least 8 different iPS cell lines, and we can always get pure CD31 positive cells. Now we take these cells and we 3D bioprint them to make this vascular network. So the way this 3D bioprinter works is it is an image of a printer. It has 2 needles, and these needles can move in a very precise X, Y, and Z orientation. And you can code how these needles would move. And one example is shown here. We're making a zigzag pattern with these needles. And what they are extruding is what is called a bioink. So bioink contains hydrogel and cells that you want to print. In this case, it's endothelial cells and other support cells of vessels, pericytes and fibroblasts. And we make a zigzag pattern. So the advantage of this is the bioink is that it keeps the cells together for a while, that they would start to interact such that they could make vascular network. And then the empty spaces in between the vessels is where the angiogenesis would happen and smaller vessels would form. So how we do it is, again, we start with our scaffold that I showed you previously. We flip the scaffold on one side. We bioprint the bioink on one side of the scaffold. We flip it upside down. We seed RP cells, where I already shown you. And then we let this tissue mature for a few weeks. And it takes about 4 to 5 weeks, but the scaffold in this case completely degrades. And the 2 cell types on both sides, RPE cells on one side and fibroblast and endothelial cells on the other side, make enough extracellular matrix to replace the scaffold with what looks like really a membrane of the back of the eye, Brooks membrane. And the tissue starts to look like this. This is a bioprinted tissue where on top you see RPE cells. In between the space where you don't have any cells is where the ECM has filled in. This is a Brooks membrane equivalent. And then here in the bottom are the capillaries labeled with endothelial cells in green and pericytes in red. And you can see that capillaries that go across the entire tissue. If I run a Z section through the bottom part, this is what you would see. Here's the capillaries of various diameters, and here's the one that is going across the entire tissue. If you notice the scale bar, the diameter of this capillary is somewhere about 20 to 50 micron. Really, what is the diameter of capillaries naturally? And the other thing is, the label here is in green is endothelial cells, and in red is pericytes that, as you can see, nicely wrap themselves around the endothelial capillaries to support them functionally and structurally. So when we put the RPE on the other side, RPE cells nicely mature, and you can see in green is a apical marker for RPE cells, which is present in this apical structure is called acerine. and in red is a marker for tight junctions. And on the bottom side, these again marked here in red are the vascular network that has nicely now covered the entire tissue. In this case, the vascular network is stained for a protein called PLVAP, which is a structural protein for what is called fenestration. So fenestration is very important. What is fenestration? Fenestration is actually in capillaries are very tiny pores, 70 to 100 nanometer pores in the membrane of endothelial cells that allow easy flow of macromolecules. Remember I said RPE cells maintain the outer blood-retinal barrier. So the nutrients have to flow from capillaries in through the RPE into the photoreceptors. And to allow that easy flow, the capillaries of choroid have fenestration in them. And that allows macromolecules to pass the RPE, the choroid and the RPE much better. So we asked when we make this capillary network with RPE cells, do they become fenestrated? Do they become choroid-like? And the answer is yes. On this side, you see what is published fenestration images from fetal human choroid published last year. And you can see these arrows mark this kind of tiny little pores in the membrane of endothelial cells. Compare that to what we see in our iPSC-derived choroid. We have a very similar fenestration network in our capillaries. So we think these capillaries, just like what happens in the choroid, become fenestrated. And we have evidence now that this fenestration is dependent on the presence of RPE cells on the other side. So here I'm showing you two examples. Here's a tissue that was printed in the presence of RPE cells and a tissue that was printed without the RPE cells. So you can notice RPE cells are present here, but they're missing here. Now what you will see is on top panels, notice the thickness of these vessels and how much they're proliferating in the presence of RPE. They're much thicker, they're proliferating more, and they have a much higher expression of this fenestration marker, PLVAP, as compared to when RPE is missing. And this is a small piece, about 100 by 100 micrometer. But if you look across the entire tissue, and we can print now tissues that are 6 by 6 millimeter, you can see in between our printed area, there's a really minimal angiogenesis that happens in the absence of RPE. But in the presence of RPE, the vessels proliferate, and as they proliferate, they become fenestrated. They start expressing the fenestration marker. So we think in this 3D tissue, RPE cells really help vessel proliferation, just like how they should do in the eye, and also help these vessels to become fenestrated. So right now, we're using this tissue for studying how really AMD pathogenesis happens in a dish, at least. And then we hope that actually we can also turn this into perhaps a tissue therapy one day where we could transplant both RPE and choroid for advanced stages of macular degeneration. So with that, I want to thank the team that really worked together for all the cell therapy project, because as you can imagine, the cell therapy project, it takes a village to do something like that. You need people with stem cell expertise, GMP expertise, preclinical expertise, and these are all the people with various expertise that came together and made it happen. And because of that, they last year got an award from Dr. Collins for their efforts to develop cell therapy on the NIH campus. So with that, I'm happy to take any questions you have.
Very exciting. Do we have some questions or comments? Is there a way that you can tell whether the animal has restored vision?
We can ask them to read. No, unfortunately, in pigs, it's a very difficult experiment to do. You can do such an experiment in rats, where we put them on a platform and we rotate a cylinder around them that has black and white stripes. And just if a rat can see, it tracks the stripes. But if the rat can't see, it will just randomly look on both sides. So we performed that experiment using our cells, and after transplantation, we can see that clearly those rats start. So these are dystrophic rats where the RPE is dysfunctional, and over time the retina starts to degenerate. So normally, if they would degenerate, they are randomly looking on both sides. But once we transplant our patch, they start to track the stripes a little better. So that's the only way we can see that we can know that they see. Pigs is much harder to do any of such experiments. So we rely on those electrical measurements of the eye.
I mean, in patients, you use laser
In patients, there are many ways to do this. Yeah, no, patients is you can just ask them to read. So it is. Say that again.
What about an anesthetized pig?
So these experiments that electrical measurements that I showed you from the back of the eye are done on anesthetized pigs.
Yeah.
But that's about what you can do looking in the back of the eye.
I'm not sure I missed this or not, but when you made that incision and put the patch in, what did you close the incision afterwards, or?
So the retinal incision is not really closed. It closes by itself. So there's a little glue, surgical glue that you put, and then when your two edges come together, what you do is you flatten the retina on top, and then the two edges come together, and over the next few days, it heals by itself. You, of course, have to suture the eye wall incision because that's a pretty large incision too. Otherwise, it starts bleeding. Yeah.
It's really beautiful work. So the question is whether or not you've been working on trying to speed up the differentiation, and what is the time frame of the RPE differentiation in the dish compared to during development? I mean, is that just the way it's going to be, or do you think you can make it faster?
So this was the fastest we could get it. And this is, so traditionally it takes about 20 weeks. So we shorten it to about half. And this half is, so RP in if you think about human development, by day 21, actually RP cells already start to be there, and then they mature over the next few weeks. So the timeline is pretty similar to the human timeline. We may have fastened it a bit using our approach, but I don't think we could, we could not fasten it anymore without compromising the quality of cells. The traditional methods that were used in some of the other studies which have used what is called a spontaneous differentiation, that takes about 20-25 weeks. That's a much slower process, even slower than what happens in human development.
What causes those capillaries to become fenestrated?
That's $1 million question that we're trying to answer now. What is it? So previously it is thought that it was just a VEGF, but nobody knows. So now we're hoping that we can kind of understand what factors RP cells are secreting to really make this fenestration happen. So we're trying to isolate those cells and see what are they secreting on the basal side. Yeah.
I can tell you then in hepatocytes, the fenestrated endothelium is actual contractile.
Okay.
The ring, which is an actin ring, is capable of contraction and modulating the size of the pore from as little as around 20 nanometers up to 150 or so.
Oh, that's interesting.
Well, any other questions? Okay, well, thank you both very, very much.